Welcome back to the deep dive. Today we're going small, really small. We're embarking on a deep molecular tour using our sources to really get inside the cell. It's like this tiny integrated factory that, well, defines life itself.
It really does. And
Yeah.
this is a great chance to, get comfortable with the specific language. If you want to understand this stuff, the terminology is key. Our sources are clear. The cell is the fundamental unité morphologica, functionale genetica of life. Think of it in terms of levels. It's the fifth level of biological organization, more complex than organelles, but the building block for tissues.
So we're really looking at the blueprint here. Where do we start? What are the sort of main structural parts we need to know?
Okay, fundamentally, you can break it down into three core components. There's the outer boundary, think of it like border control, that's the membrana cellulara or plasmalemma. Then you have the whole internal environment, the factory floor, which is the cytoplasma. And finally, the control room, the boss's office, the nucleole.
Okay, let's start with that border control, the plasmalemma. It's not just a bag, is it? It gives the cell its shape and keeps the inside separate from the outside, the medial extracellular?
Absolutely. It's much more than a bag. Its function comes directly from what it's made of. Primarily, it's phospholipid and a whole lot of different protein. And interestingly, the outer surface isn't smooth. It's coated with negatively charged glucide things like glycoproteins and glycolipids. They're involved in communication.
Right, and the structure itself is, well, it's pretty famous, the modello-mosaic fluid. That really describes how the phospholipids behave, doesn't it?
It does, exactly. The phospholipids naturally arrange themselves into this double layer, the bistratlipidic. Imagine two layers of molecules with tails pointing inwards. These tails are hydrophobic, water-hating. So they create this oily, water-resistant core right in the middle of the membrane. A mise hydrophobe.
Like, hydrophobic core, that sounds like a barrier. Why is that important? Why care that it resists water?
Because that core is the barrier. It stops ions and water-soluble molecules, molecule hydrosoluble, from just flowing straight through. If everything could just pass freely, the cell couldn't maintain its internal balance, its specific chemical environment. And crucially, it couldn't maintain its electrical charge difference, the potentiel de membrane.
Ah, the membrane potential. So the membrane isn't just a physical barrier, it's electric.
In a way, yes. It maintains an electrical gradient. The cell actively pumps ions across the membrane to keep different concentrations inside versus outside. This creates a voltage, like a tiny battery. It's fundamental, especially for nerve and muscle function, but really for all cells.
Okay, so to control that charge and let specific things through nutrients in, waste out in these gates, right? That's where the proteins come in. That permeabilitates, like a Mia idea.
Precisely. Those proteins are the gates, the channels, the pumps. They allow for shimbidirekinol, a two-way street. Nutrients come in, waste products, proteolytic catabolism go out. But it's all regulated.
And our sources talk about different kinds of transport. There's passive stuff like diffusionia and osmosa, just following concentration gradients. Easy enough. But then there's the hard work. Transportal active. That sounds like it needs energy.
It absolutely does. Active transport means moving things against their gradient, like pushing something uphill. This requires significant energy input, cellular energetica, usually in the form of ATP, and it uses specialized protein machinery, often called pumps, to physically move substances across.
Okay, let's move inside that boundary now into the cytoplasma. You called it the factory floor. It sounds busy.
It is. It's the cytiole, the main location for most of the cell's vital functions, funci vitali. Structurally, it's described as a system colloidal. Basically, it's mostly water, the medial de dispersi. And suspended in that water are all sorts of molecules and tiny structures, the mycelial colloidal.
And this cytoplasm isn't just one uniform goo, is it? There are different parts.
That's right. There's the fluid part itself, the unstructured bit, called the hyaloplasma or cytoplasmic matrix. And then embedded within that, you have the structured components, the organelles cellulare, the cell's little organs or machines. These can be common ones found in most cells or specific ones for specialized cells.
Right, let's get into those common organelles, the actual utilities, the machinery. Let's start with the reticular endoplasmatic, the RE. What is this thing? It sounds like a network.
It really is. Think of it as a vast network of interconnected membranes, a system canalicular. It actually stretches from the outer cell membrane, the plasmalemma, all the way to the membrane around the nucleus. Functionally, it's like the cell's internal circulatory system, the system circulator intracytoplasmatic. And there are two types.
Okay, two types. There's the rough ER, RE rugose. What makes it rough, and what does it do?
The rough comes from tiny dots studded all over its surface. Those are ribosomi. Because of these ribosomes, the RE rugose, which is also called ergastoplasma, is heavily involved in making proteins, synthèse de protein, specifically proteins that are going to be shipped out of the cell or embedded in membranes.
Makes sense. Ribosomes make proteins, so rough ER is protein central. What about the smooth ER, the RE needed?
No ribosomes, so it looks smooth. Its job is different. It's crucial for synthesizing lipids, fats, and it plays a really important role in breaking down glycogen, metabolismal glycogenuli, especially in liver and muscle cells. Detoxification, too, in some cases.
You mentioned the ribosomes. Ribosomi, tiny dots making proteins, also known as corpusculi louis-palladi.
That's their historical name, yes. They are incredibly important little machines made of ribonucleoprotein. Tiny granules, really. Whether they're attached to the ER or just floating free in the cytoplasm, they are the site, the sigil of protein synthesis, synthese protease. They read the genetic instructions and build the proteins.
Okay, proteins are built. But all this building, pumping, moving, it takes energy. That brings us to the power plants, the mitochondria.
Ah, yes, the
Yes.
mitochondria. Absolutely essential. They have complex structure, a double membrane wall, technically trilaminar, made of lipoprotein. The inner membrane is the key part. It's highly folded into these structures called crest mitochondrial. These folds massively increase the surface area inside within the internal space called the matricia mitochondriala.
And why is that surface area so important?
Because that inner membrane is where the main energy production happens. Mitochondria are the CDO of phosphorylation oxidative. This is the process that uses oxygen to break down fuel molecules and generate the vast majority, I think around 95% of the cell's ATP. It's energy currency. Without mitochondria working properly, the cell basically runs out of power very, very quickly.
95%. That's staggering. Okay, so proteins are made, energy is generated. What about packaging and shipping? Is that the Aperdal Golgi?
Exactly. The Aperdal Golgi, sometimes called dictyosomy, is like the cell's post office or finishing department. It's a system of flattened sacs called cistern, plus little transport bubbles, micro and macro vesicles. Its main job is to modify, sort, and package proteins and lipids that come from the ER, preparing them for delivery, often for excrea or secretion outside the cell.
And every factory needs a waste disposal and recycling system, right?
Absolutely. That's the role of the lysosomy. These are small spherical sacs filled with powerful digestive enzymes, enzyme heterolytis. Their job is digestia intracellular, breaking down worn-out organelles, debris the cell might take in, or even bacteria in some cases. They're really critical in immune cells, the cellulae phagocytari, that engulf and destroy invaders.
One more common one, the centrosomal. This one's involved in cell division.
Yes, the centrosomal is key for cell division in animal cells. It usually sits near the nucleus. Structurally, it's got two small cylindrical structures called centrioli, arranged at right angles to each other, inside a cloudier region called the centrosphere. It organizes the microtubules that help pull chromosomes apart during division. But interestingly, it's absent in mature nerve cells, which is a big reason why they generally don't divide.
Okay, that covers the main machinery. Now let's get to the command center you mentioned earlier, the nucleus. The nucleus.
Right, the nucleus. It's arguably the most important organelle. It has several crucial roles. It coordinates the cell's fundamental activities. It houses the cell's complete genetic library, the material genetic. It controls the cell's overall metabolism, metabolismal cellular, by controlling which proteins are made. And absolutely critically, it's responsible for passing on that genetic information, transmitting informai genetis, when the cell divides.
And looking at nuclei, it's not always one per cell, is it? Our sources mention some variations.
That's a fascinating point. Most cells are indeed mononucleate one nucleus, but you find exceptions. Liver cells, hepatocytes, can sometimes be binucleate with two nuclei. Skeletal muscle fibers are typically polynucleate, containing many nuclei within one long cell. And then there are the really specialized cases like mature red blood cells, hematia, which are anucleate. They actually get rid of their nucleus to maximize space for carrying oxygen.
Wow, no nucleus at all. Okay, so we do have a nucleus. What's inside? What's its structure?
Well, first, it has its own double membrane, the membrana nucleara or envelopa nucleara. It's trilaminata, like the cell membrane. And it has pores, nuclear pores, which regulate traffic in and out. And interestingly, the outer layer of this nuclear envelope is often continuous with the rough ER, the RE cytomembrane, and even has ribosomy on its surface. The space between the two nuclear membranes is called the spiule paranuclear.
And inside that envelope?
Inside is the karyoplasma, which is like the cytoplasm of the nucleus, a kind of gel-like substance. Suspended within the karyoplasma is the chromatina. This looks like a tangled mass of threads most of the time. But when the cell gets ready to divide, this chromatina condenses, coils up incredibly tightly to form the visible structures we know as chromosomai, the chromosomes.
And those chromosomes, the condensed chromatin, that's where the genetic information actually is.
Right.
What are they made of?
Exactly. Chromosomes are incredibly complex structures. The main component is, of course, ADN, DNA, but it's not just DNA. It's tightly packaged with various proteins, especially protein histidinase, which help coil it up, and protein non-histidinase, which have other regulatory roles. There's also some ARN, chromosomal lipids, and even ions like calcium, texme2+, and magnesium involved in maintaining the structure. It's a very organized package.
Incredible complexity. So just to round things off, we've talked about common organelles, but some cells have specialized extras, right? And all cells share some basic abilities.
Yes, absolutely. Specialization often means developing unique internal structures. Muscle cells have myofibrillae, the contractile protein bundles. Neurons have neurofibril for structural support and helping with nerve impulse conduction, conduceria impulsului nervos. Neurons also have corpuscle, which are basically clumps of ergastoplasma, rough ER, focused on making neuron-specific proteins.
And the universal functions. What can pretty much every cell do?
Well, generally speaking, all healthy cells carry out synthesa proteica, a protein synthesis. They manage metabolismal cellular energy and substance conversion. Most have the capacity for reproduceria cellulara, cell division, although some, like neurons, lose this. They all perform transportal transmembranar across their membranes and maintain that crucial potenziale de membrana, the electrical charge.
But specialization takes these basics and pushes them further.
Precisely. You get proprietae speciale. Muscle cells develop high contractilitatea, turning chemical energy into movement. Gland cells develop activitatea secretory to release hormones or enzymes. And nerve cells excel at excitabilitatea conductibilitatea, the ability to generate and transmit electrical signals, the potenziale de acciune.
It's just
Yeah.
an amazing level of organization and integration. From the outer membrane to the DNA packed in the nucleus, every part seems to have a precise, vital role. It's not just a bag of chemicals, it's a dynamic factory.
It truly is a marvel of efficiency. And maybe the key takeaway linking it all is that energy flow. Remember the mitochondrial generating maybe 95% of the ATP through phosphorylarea oxidativa. That ATP fuels almost everything else we've discussed, the pumps in the membrane, the protein synthesis, the movement.
Right. And that actually leads perfectly into a final thought for you, our listeners, to chew on. We've talked about how complex transportal transmembranar is, maintaining that potenziale de membrana, using active Pompe working against the gradient, the Tunson tri, all thanks to that permeabilitate selectivar. So here's the question. What happens to that delicate energy balance, that membrane potential, and maybe even the cell's ability to function or communicate, if its mitochondria suddenly can't make enough ATP? If the power plant goes offline, how long can the border control keep working effectively? Something to think about.